Good afternoon, learners. Welcome you all again to this one of my next sessions. If you remember, in my last sessions, I actually dealt with BBYET 143, that is Economic Botany and Plant Biotechnology, and already I had quite a number of sessions on several units of this particular course. Now today I will take up another topic, that is construction of DNA libraries and genetic engineering methods. And this is also a part of this course, that is Economic Botany and Plant Biotechnology. And you will find most of these discussions in your unit, that is recombinant DNA technology. I already dealt quite a number of topics on recombinant DNA technology in my earlier sessions, but I thought that some of the topics might need to be elaborated in order to make you better understand. And therefore, today I have taken this topic which belongs to this unit, that is recombinant DNA technology. Now, you will see from today's title that actually there are two components. One is construction of DNA library. So library is one component, and the second component is genetic engineering methods. So there are two topics which need to be discussed, and I will try to divide the time accordingly and discuss or make you understand these two particular topics. Now, to start with, what is meant by a library or a gene library? Now, all of us know what is meant by a library, because you know that when we go to it can be a book library, it can be a video library. So library is a place which consists of a collection of certain things. If you go to a book library, then you find a collection of books. If you go to a video library, you will find a collection of different videos or CDs maybe. Then what is gene library? So the concept is that a library is such a concept where you actually have a collection of all genes. Let's take the example of any organism, human or any animal or plant, like let's say Arabidopsis or rice. If somebody asks you the question that what are the genes which are present in rice? I mean, it's very difficult to answer this question because there are so many genes. How many genes are there in human, and what are those genes which are present in a human system? The answer is it's a huge number of genes. So you should have some component, or you should have some way out by which you can actually mention, or you can actually describe the gene pool which is present in an organism. And that is what a library means. It's a collection of genes. Now, obviously, we need to prepare this library. That is, experimentally, you need to construct a library. It's not ready like you construct a book library. You have to construct it. You have to go for the collection of books. You have to keep them in proper order. You have to catalog them. So similarly, here also you have to create, and that is through proper experimentation. You have to create a library. Now, when we are talking of a gene library, then there are actually two components. One is it can be a genomic library, or it can be a cDNA library. What is the difference between the two? That is, when you are talking of a genome library or genomic library, you are dealing with the entire genomic component. That means in a genome, there are both exons as well as introns. Exons which are actually expressed, and introns which are actually spliced out during RNA splicing. But when you talk of a cDNA library, you don't have the intronic sequences, because you get the cDNA from the RNA. Because when we prepared the cDNA, we actually prepare it from RNA using one particular enzyme, which perhaps all of you know, that is reverse transcriptase, for which Nobel Prize was awarded for the discovery of reverse transcriptase. So you construct the cDNA library, and from the cDNA actually you try to clone the cDNA. Like you clone the genomic sequences to construct a genomic library. Similarly, you clone the cDNA sequences to get a cDNA library. But in any case, they are actually different forms of gene library. So my first slide or my first PowerPoint presentation will show that what is meant by a gene library. It's a random collection of cloned fragments in a suitable vector that ideally includes all the genetic information of that species. And this is also called the shotgun collection. There are two ways by which gene libraries are made, referred to as complementary cDNA and genomic DNA libraries. Genomic libraries and clones are necessary in addition to cDNA clones because cDNA are generated from processed mRNA which lack introns and other sequences surrounding the gene. As I said that the cDNA, they only represent the exonic sequences, that is the expressed sequences, whereas the genomic sequences, they represent both, that is the exon as well as the introns. So there is a basic difference between the two. The genome is remarkably complex and a particular fragment comprises only a small fraction of the whole genome. Therefore, construction of a useful and representative recombinant genomic library is dependent on the generation of a large population of clones. So I hope you got the idea what is meant by library. So library, now if I define it in a better way in terms of molecular biology, then a library actually represents a series of clones. So what you have to do is, let's say if I ask you to prepare or create or construct a genomic library from rice or Arabidopsis, obviously you have to understand that you have to start from the genomic DNA. So you have to first obtain the genomic DNA or chromosomal DNA of that particular organism, whether rice or Arabidopsis, fragment it, that is, you digest the DNA, okay, partial digestion of the DNA using certain restriction enzymes, restriction endonucleases, and then you have to clone it. Normally, for cloning, we use vectors like plasmids, cosmids, bacterial artificial chromosomes, yeast artificial chromosomes, and so on. There are so many vectors. Bacteriophage can even be used as a vector. So there are so many vectors, and each of these vectors, they have their own capacity. Like the plasmids, they can accumulate smaller fragments. Cosmids, a bit larger. If you go for bacterial artificial chromosomes or yeast artificial chromosome vectors, they can accommodate even larger fragments. So when you are dealing with a genomic library and you are isolating the genomic DNA, digesting it with restriction enzymes, what is your expectation? What do you expect to see? You expect to see a heterogeneous group of fragments of different sizes, some of which may be large size fragment, some of which may be small size fragment. But mind that all these fragments need to be cloned. I already discussed about the cloning procedures in my earlier sessions. What are the steps for cloning? I have already discussed. If you have followed that, then now what you are doing is, the difference is that here you are not cloning one particular fragment, but you are cloning a series of heterogeneous fragments. So it's a more kind of random process. And not only you are not using the plasmid vector, because the capacity of a plasmid vector is much less, because some of the fragments can be huge, large size fragments, and for that you need to use some vectors with high capacity. So they can be bacterial artificial chromosomes or yeast artificial chromosomes or maybe cosmids. So such kind of vectors need to be used to accommodate the heterogeneous fragments generated as a result of restriction digestion of genomic DNA. And then you have to select those clones, and then you have to identify or sequence those fragments and see what it is. There are several ways by which